guys probably have all figured that out. It depends, of course, on some of them have been cut in more ways, but we have a frontal lobe, parietal lobe, in the very back, occipital, and then on the side here, temporal lobe. Longitudinal cerebral fissure is the big groove right down that sagittal line. Cerebral hemispheres, each half is a hemisphere. The cerebral cortex is, I have one that's been cut into the lobe so we can see, can you see the gray matter? Here, that's the cerebral cortex. You guys remember what kind of stuff you find in gray matter? Why is it gray? It's no, that'd be the white. Oh. It's unmyelinated. That's where cell bodies are located, and in unmyelinated axons. The white matter is where we have myelinated axons running. So just this outside folded edge, that's gray matter. That's the cerebral cortex. In the cortex, we see obviously all these folds. So the raised part of the fold, the ridges, are called gyri, or gyrus is singular, and the grooves in between them are the sulci, or sulcus, is singular. Olfactory bulbs, we did have, I think, one, maybe or two that showed these all right. On the underside of the frontal lobe, you'll see a tract, a nerve tract, and then the enlarged tip. That's the bulb. And the little tiny olfactory nerves would go into your nasal cavity from there. This one, we can sort of see that lighter colored tract and that end. It's a little frayed, but that's an olfactory bulb. Corpus callosum, you're going to see from a sagittal view, it's that thick, white, tough band of tissue here. Just when you get through that longitudinal fissure, that's where the right and left hemispheres would be connected. So in different cuts, you might see it a little bit better. So it's that top ridge of white tissue. Lateral ventricles would be the hollow space inside in each hemisphere. There is a little bit of a wall that you would have to get through to see into that hollow space. So the hollow space is a lateral ventricle, and that wall, and some of them it's a little bit more clear, that is the septum pellucidum, the pale wall, septum pellucidum. So all of that is part of a cerebrum, also known as the telencephalon. Now for the diencephalon. Diencephalon is deep to cerebrum. It's got several major parts, and the very middle of the diencephalon has fluid in it, and that fluid-filled space, I'm going to show the model here, so we can see the diencephalon here. When they're together, that middle space that would be filled with fluid is the third ventricle. The tissue around, the main portion of the diencephalon, all that tissue here, all this tissue here, that is the thalamus. In the middle of the thalamus, and it depends on the cut if we can get a good one, but there's a little bit of tissue that sticks out that would connect the right and left halves of the thalamus. That is the massa intermedia, or you could say intermediate mass. But it's right in the middle of the thalamus. Pituitary gland is not present on the calf brains, but we can see it on our model. It's this little gland that hangs down. And so that's pituitary gland. It comes from the optic chiasm, which are where the two optic nerves cross each other. And we can see optic chiasm here. So from here and here, there would be optic nerves going out to each eyeball. This is where they cross tracks. So optic chiasm. Those are parts of the diencephalon. I don't have hypothalamus on the list, even though it is important, but there's often not a lot there to see. It's just a little bit of tissue between the thalamus and the optic chiasm. For mesencephalon, that's also known as the midbrain. You have the cerebral peduncles, which are these tracts of fibers that are running here, connecting the brainstem with the cerebrum, so it's called cerebral peduncles. On the back side of the mesencephalon, we'll see 4 bumps. So these 4 bumps here, the 2 big ones that almost look like a baby's bottom, and then 2 smaller ones below, All 4 bumps together are called the corpora quadrigemina, and there's some eye and ear reflex centers that are located there. Metencephalon, also known as basically the pons, the biggest bulge that we see, and the cerebellum behind it. So pons is the bulge. Cerebellum is the big folded structure behind it. In cross-section here, sagittal view, you can see some of pons here, cerebellum here. There's a bit of fluid-filled space. If I can hold that down there, a little bit of fluid-filled space. In between those two, that's the 4th ventricle, so just where CSF would form right in there. Cerebellum, here you can see the white branching, almost tree-like pattern. That's called the arbor vitae. So in all the sagittal cuts we can see arbor vitae. And then folia are the names for the folds that you would see on the surface. So almost like the gyri, but they're a little bit more delicate, small. So folia. And then the vermis is the name we use to describe that middle piece, separating the 2 halves of the cerebellum. So vermis on these brains sort of stick out a little bit, that prominent ridge. On this human model, you can see it as that middle piece. So that's vermis. Then the myelencephalon, also that part of the brain is known as the medulla oblongata. So depending on the cut we get, it's just below the pons. So it's hard to see the bulge here, pons, and just below that, medulla. Let me see if I can find one that's got a little bit longer. That's probably as long as we get. All right, so pons, the big prominent bulge, a smaller bulge below it, medulla, and then when it narrows, that becomes the spinal cord. The spinal cord you can see here. There is, depending on the cut we get, a hollow tube running through the spinal cord, and that's the central canal. I'm not sure we have that very well. All right, those are basic parts. Now, for the sake of the test stuff on next week, I just wanted to say, for a lot of you, I have to come up with 3 to 5 questions for however many of you there are. So there might be a few repeats. I might even ask a few kinds of function questions. And again, raise your hand if you know it. Don't raise it if you don't. But you might want to do just a little bit of review, or white matter versus gray matter, some really basic kind of things there. So just to come up with enough to ask the class to get all those questions answered. So any of the structures are fair game. I will ask just a few things on the plastic model as much as possible. I want to use the real thing because obviously you've already taken a class where you've studied models. Mm-hmm. This is the class where you study the real thing. So I want to do that as much as possible. But a few things like pituitary gland we got to use the model for. And the eyeball, we'll do eyeball too. So next week, you guys will come. I might not be able to get here until more like 15 or 20 till, but there's no setup or anything. So we'll just spend some time reviewing, and then once it seems like everyone's had their chance to review and we're ready to go, I'll gather you around like this and get the eyeballs out and a few of the brains out, and I'll start pointing and having you guys name.